reproductive biology, and I didn't realize that much that I was going to be doing reproductive biology on worms. But I don't know, I love it. It's fascinating to me how something can survive like that. I love things that survive, whether it's plants or animals or worms. I just love things that just, so resilient. Yeah. Well, actually, these guys, of all the breeds of sheep, these are probably, these guys are resistant to worms. They do well in a lot of different environments. They shed that hair, so they don't need a whole lot of help. So as far as sheep goes, these are probably hardier than most breeds of sheep. So, and the biggest concern we have is Cumulus contortus, or barber pole worm. You actually can see that worm when you go back to the lab. It is a worm that sucks blood, and it can suck so much blood it leaves the animal anemic and death. The life cycle of this worm, complete life cycle, takes about 5 weeks. So it can build a new generation in one summer. So from generation to generation, this worm evolves so quickly, and that's why resistance evolves so quickly. This is the most amazing worm. It used to be just a tropical worm, and now we find it as far north as in Maine and Canada. This worm has probably adapted to climate change, so it can evolve. The genetics of this worm can evolve so fast that we can't even keep up with it. There's probably different strains of the worm all over the U.S. So what we do is we find ways, a number of different ways, to help control the parasites. So we can actually breed from more resistant animals. And in fact, there's a sheep program that helps farmers to understand the flock, understand genetics. So that's right now, that's probably the most promising way that we can control parasites is through genetics.
So are they blood testing that? How are they?
They're trying to develop a blood test, and because the genetics for resistance is controlled not just on one loci, but several, it's really hard to isolate. We sent some DNA samples up to the Beltsville lab, the ARS Beltsville lab, and we're waiting results on that. We've had everything from really resistant fires that we sent to really susceptible. So what you notice around here, we're now rotationally grazing these animals, so we take advantage of excess forage and move them to where there's good excess forage. So we'll have a group moving over there. And by moving the animals around like that, you're spreading the parasite problem. You're kind of minimizing the parasite problem by moving the animals.
Do you sell the males?
Yeah, being a federal agency, anything that we sell has to go for public bid. We send the really good ones to what's called the Cotton Expo. We don't really want to compete with producers. That's not our goal. But we want to offer good genetics to producers. So we work with the other producers that have something similar. If they don't want it there, we're not there. If they want the product there, then we'll offer it. So we kind of work with the other farmers to make sure that we have a niche going on.
The only way we've had of putting poultry litter on has been to spread it out on the surface. That's because it's a very bulky material. Now, if we were in a tilled system, like with row crops, what you could do is you could put it out there, and then you go through with a disc and you kind of turn it under. That would work okay. But in pastures, of course, these are perennial pastures. We don't want to destroy our grass, so all we do is just throw it out on top and let it hopefully soak in. By doing that, we leave it completely exposed to the atmosphere. And two things are going to happen that will cause problems. One is about half your nitrogen is going to evaporate into the air as ammonia. The other thing is if there's a big rainstorm, if you've ever been near a field where litter has been spread, you know the water coming off there will look kind of like coffee because of all that organic matter that's leaving. And along with it, a lot of the nutrients are going too, including phosphorus. If those leak off into the streams and lakes, then you get these algal bloom problems and water quality problems. And of course, there's another factor that a lot of people are concerned about that's from a farming standpoint, it's not a big problem, but for neighbors, the smell can be pretty bad when litter is spread. So we'd like to eliminate all of those problems as best we can. So what we're looking at is trying to figure out ways of preventing those nutrient losses from pastures. And because poultry litter is the big pasture fertilizer around here, we've especially focused on trying to find ways to use poultry litter more efficiently. Keep the nutrients on the crop where the plants can use it, but not allow those to escape either into the atmosphere or down into the streams and lakes. Now, in the case of this study here, it won't deal with the problem of the smell, but we do have some various best management practices going on here trying to prevent the losses in runoff. What we'd like to do is just get it completely under the surface, trapped away from the atmosphere. But of course, that's not this study here. This is one strictly on best management practices for grazing, various ways of grazing the animals or not grazing it, keeping it in hay. Took quite a bit of time and investment to set this up, and it's a long-term study. This one's set up for 20 years. I think we're in about year 11 now on this. And so we're testing all of those various ways of trying to prevent nutrient losses from the field. And over time, every year we can collect every runoff event that happens on these, compare the runoff from those various treatments, see which ones lose the most water. We take samples of that to the lab, look at how much soil is in that, how much sediment's collected, how much phosphorus is being lost, how much nitrogen is being lost. And we also do some soil sampling in the fields here to check for soil compaction, for instance. So this data from this will be used by the Natural Resources Conservation Service in a lot of their recommendations. I can tell you we have other work going on where we're actually experimenting with putting poultry litter under the surface of the ground in pastures as well with a machine we're developing that we call the subsurfer. Okay, well, we can head on back up the road as we go. If you haven't looked at these plots, if you would look and see what I'm talking about on the various streets.
We've been doing research with the parasites for about 8 years now, something like that. So I have looked at a lot of fecal samples, we've done a lot of blood work, and we've cultured a lot of worms. So we can both talk about parasites for hours. So this is what we do all the time. We can tell how wormy an animal is by the color of the tissue in their eyes. It's called phaumatosporin. Also, another way the ultimate way to tell how wormy an animal is by running the packed cell volume or the hematocrit in their blood. Here's my gross picture. These are actual worms. This is the stomach or the intestines of a sheep that has died from worms. Each one of these worms, the Homunculus contortus, it's got a little hook on its head, and it attaches to the lining of the intestine, and it sucks blood. Each worm can take 1/10 of a ml of blood per day. And if they have thousands of worms, it takes their blood away real fast and it kills them, and that's the problem with the parasites. So we individually deworm, and that's another management practice. You don't worm the whole flock at a time. You manage it by individual animals, deworming them. So this is what we've worked on for many years, and now we'll get to the fun part, and that is how do we analyze for the worms. We use the fecal egg count, we use the hematocrit, and we use phaumatosporin, and we can pretty much keep an eye on the whole flock as to how we're managing the worms and everything. So now you're going to get to find out how we do fecal egg counts. So come on around. You guys are going to get to actually do the mixing and everything. Okay, you're going to take 2 grams. I'll let you use this. You don't have to pick it up with your hands or anything. Okay, here's our salt. You're going to get 28 mils of salt water. You see that 30 over there? Okay, there's 2 marks below it. That's 28. That's where you want salt. Okay. And you take 28 mils. Okay, now pour that through there. Kind of tip it up a little bit like this, hold on to that, and smush it around into that solution. And you can come over here and weigh your